Hi, Professor. Can you hear me?
Hello, hi there.
Uh,
How are you? Hi.
Hi. Hi, good morning. We are very glad that you have accepted our invitation and would like to join us with today's sharing. So before we start today's sharing, could you please give the audience a brief introduction about yourself and also the research you are focusing on?
Yes. I am Claudio Mauro. I'm a professor of metabolism and inflammation at the University of Birmingham in the Institute of Inflammation and Aging. And our work really looks at the relatively new field of immunometabolism, which is how metabolic pathways of cells and tissues really control the immune response. And now, in exchange, the immune response um, impacts the homeostasis and the pathologies of tissues in our body. In particular, we focus on some metabolites as signaling molecules. I believe we were among the first to suggest immunomodulatory molecules for the metabolite lactate, but we also have another interest in the signaling properties of saturated fatty acids, particularly in conditions like obesity and diseases associated with obesity.
So, since today's topic is inflammation and aging, right? So, not all of our audiences are professional medical personnel. So, to begin with today's conversation, we may start with some basic concept. Could you introduce to the audience about the definition or the meaning of inflammation and what will truly trigger inflammation?
Of course, I mean, inflammation is a body response to an insult. The lay public generally associates inflammation to an insult due to a pathogen, such as a virus or a bacteria. But actually, the insult can be broader than that, such as stress, burnt, trauma, and so on and so forth. And inflammation can be divided broadly in acute versus chronic. An acute inflammatory response is a response of the body to an insult, which dissolves within a couple of days, whilst a chronic inflammation can last for weeks, months, and even years.
So, like the chronic inflammation is also taken as a hallmark of aging nowadays. So, Professor, in your opinion, what is the relationship between inflammation and aging, and how do you understand the term inflammaging?
How should we understand inflammaging? I think inflammaging is a chronic low-grade inflammation that happens in the body and establishes generally with age and leads to a wealth of conditions that is induced by chronic diseases. At the same time, this low-grade inflammation can lead to accelerated aging.
So, I've heard that your study had been focusing on immunity, right? But inflammaging is also an extension of immunosenescence. So, how do immunosenescence and inflammaging related to each other, and what is the difference between these two concepts?
I think actually inflammaging promotes senescence of immune cells. If you can see that. So this low-grade chronic inflammation that happens with age of the human being in the body leads to accumulation of immune cells, but in turns, then senescent immune cells further promote this chronic low-grade inflammation in the body, and so promote inflammaging. So this is a feed-forward loop, which, we need to break in order to maintain a healthy immune system, even with aging.
Yeah, that's right. But like with the accumulation of those senescent cells, I believe that there might be some like diseases related to it. So what kind of age-related diseases that are associated to inflammation and how is those immunosenescence and inflammation harm our human body?
Yeah, I mean, there are a number of diseases of ages of age that are underpinned by inflammation, such as type 2 diabetes, arthritis, cardiovascular diseases that are a consequence of obesity. For instance, obesity is a known factor to promote inflammaging and immunosenescence cells. So these conditions affect the homeostatic processes of our immune system in tissues like the adipose tissue and the muscles in the first instance. And then that, I believe, is transmitted as a consequence to a wealth of other tissues in our bodies, such as the lungs, the livers, and so on and so forth. So the connections between tissues of our body via the immune system is a very hot topic of the research at the moment.
I've heard that you mentioned type 2 diabetes. So, in my opinion, it is also considered as a metabolic disorders. So, how is those metabolic disorders like type 2 diabetes or obesity? Obesity, I think in your recent research, you also mentioned that after weight loss, individuals with obesity will still exhibit an inflammatory phenotype. So, how are these metabolic disorders related to inflammation and aging?
Yes, I mean, this is very interesting. We know now that systemic metabolism can have an impact on the immune response, and the immune response in turn can have an impact on the systemic metabolism of the body. One key observation of a few years ago nowadays is how TNF, which is an inflammatory molecule, a key inflammatory molecule, interferes with the signaling of insulin and insulin receptor. So that clearly linked inflammation to type 2 diabetes, for instance. So we know that nowadays that by interfering with the metabolism of immune cells, we change the immune response. We can turn the immune response into a pro-inflammatory one or into an immune suppressive one. And that's why these areas become so interesting. And, pharma have, in the last 10 to 15 years, invested so heavily in the metabolic pathways that control the immune response, because we know that in cancer, where we would like to have more killing type of immune cells, we can use metabolic drugs or drugs that interfere with the metabolism of the immune cells to achieve more killer cells in the immune system. But at the same time, in autoimmunity, where you would like to have more immune regulatory and immune suppressive type of immune cells, again, you can intervene on the metabolism of the cells to obtain an increased number of these immune suppressive T cells. So this is the metabolism of the cell, the autonomous metabolism of the cell. But at the same time, the systemic metabolism, such as in obesity, can really push your immune response towards an inflammatory type of response. And then how do we return to an homeostatic type of immune response is a subject of intense investigation, and we are working in this area at the moment.
I've heard that you've mentioned TNF signaling pathway. I saw that in your paper there is another transcription factor, that is NFκB, which is also very important in this overall process. So how is NFκB transcription factor influence the process of inflammation, and how is it involved?
It's a great question. It leads me back to my PhD and postdoctoral studies. I mean, I haven't directly worked on an NFκB field in a few years now, but I spent the first 10 plus years of my career working on NFκB pathways. And so NFκB is a family of transcription factors that really control two responses mainly. One is the inflammatory response, and the other one is the control from cell death, and particularly from apoptosis, but also from other forms of cell death. And TNF-alpha, by engaging with the TNF receptor, is able to activate NFκB, which in turn translocate from the cytosol into the nucleus to bind promoters on the DNA and in promoting transcription of specific genes, some of which belong to the family of inflammatory genes and others to the family of anti-apoptotic genes. I mean, the mechanisms of NFκB activation downstream of these key inflammatory cytokines, such as TNF, are very complex. One important protein complex is the IKK complex, which is the IκB alpha kinase complex, which is a kinase that phosphorylates the main inhibitor of NFκB, IkB alpha, and targets it for degradation via the proteasome. And so that the dimer of NFκB is released from this inhibitory control by IkB alpha, and it's free to translocate into the nucleus where it initiates gene transcription. Of course, TNF alpha is not the only inflammatory molecule that activates NFκB, but pretty much all inflammatory molecules will lead to activation of NFκB transcription factors.
That's the course of inflammation and how they are accumulated. You have just said that obesity will lead to inflammation, but obesity, is an metabolic disorder. They will also produce some metabolic products like those metabolites. So how do these metabolites in the human body induce those immunosenescence?
Well, so one research that we have conducted in collaboration with a group in Milan, the group led by Professor Massimiliano Ruscica, has shown how lactate can accumulate in the adipose tissue, for instance, and this leads to a series of responses which lead to increased inflammatory response by the CD8 T cells in the adipose tissue. This is something we published recently in iScience, which the audience here can go and have a look at if they want to. But another area that we are studying at the moment is how in obesity an inflammatory adaptive immune response, such as T lymphocytes in particular, is achieved. And this is key because this inflammatory adaptive immune response then links obesity to a wealth of human conditions such as cardiovascular, cancer, and autoimmunity. So how does that happen? Of course, in obesity, you have increased circulating inflammatory factors, right, such as TNF alpha, such as interleukin and interleukin 6, and so on and so forth. But we are studying the direct involvement of saturated fatty acids, such as palmitates and ceramides, into turning T-cell responses into inflammatory type of responses. For instance, we have published recently, both in cardiovascular research and in biochemical pharmacology, how ceramides and other saturated fatty acids can promote an inflammatory type of T-cell response, and how in omega-3, which are unsaturated fatty acids, which you find enriched in fish oils and certain vegetables, can counteract this effect of palmitate and ceramides by promoting a sort of immune suppressive response. So we have proposed that omega-3 in obesity can tip the balance of T-cell responses towards an immune regulatory, immune suppressive response that switches off the inflammation induced as a result of obesity. More groundbreaking, I believe, studies we are trying to finalize at the moment, where we show how saturated fatty acids can directly lead to a pro-inflammatory response of the T cells. And we have mechanisms suggesting how that works, which are imprinted in our T cells for a long time. And even after we go on to lose the weight, our data seem to suggest that that there is a biasing of the T cell response towards inflammation that lasts well beyond the point of weight loss. And actually, this could be a driver of weight regain, in accordance with a couple of other papers that have been published so far. So that could suggest why individuals who lose weight then tend to regain this weight within 6 to 2 years from the weight loss. And we believe this is a very important finding, because it suggests that even if we have lost the weight, our T cells are still programmed to do inflammation, and this could impact negatively the consequences of obesity in the long run, which are, as I said, cardiovascular, autoimmunity, and so on and so forth. But we have identified a couple of pathways that we could intervene on these T cells that could promote the reestablishment of the homeostatic T cell response. And we hope this will be the subject of a publication in the near future.
So you had mentioned that like the cells have a memory and they have remembered us as an obese person, and even though we have lost some weight, they still can maintain these kind of inflammatory level. So is there any solutions right now that has been published that suggest to delete the memories of the cells and help us to lower our inflammatory level and be healthy again?
Not directly. I believe there are of course certain things that would help with that. For instance, moderate exercise, moderate but repeated exercise is one of those things that can switch the systemic metabolism towards a sort of catabolic metabolism that reduces inflammatory cells or immune-senescent cells, senescence immune cells, and so favors reestablishment of an homeostatic immune response. I believe, as we suggested in some of our publications, omega-3 could, so a diet rich in fish oils could favor that kind of immune response. And at the same time, I think some drugs, such as metformin, are known to interfere with the metabolism of immune cells, and so kind of promote a return to an homeostatic type of immune response in obesity or in aging. At the same time, there are some novel drugs, such as the GLP-1 inhibitor, that are used in the obese population and have recently been shown to potentially have a cardioprotective role. These again could be tested as drugs that promote a rejuvenation or a retardation of the immune senescence. A recent paper that I would like to highlight is a paper that has shown the effects of SGLT2 inhibitors on immune cells. And now these inhibitors of SGLT2 could be used in autoimmune diseases. This paper has appeared just a couple of weeks ago, I believe, in Cell Metabolism. So these are the things that are in my mind at the moment that are more promising, but I believe we can come up with additional targets, and I believe the promise is the immunometabolism is going to deliver a number of metabolic enzymes and or sensors of metabolites, such as transporters, which will become the next generation targets for the immune system.
So, like those metabolic products you have mentioned, like lactate, they're just so common in daily life. Even like anaerobic exercise, if we do anaerobic exercise, lactate will accumulate during this process. So, is there any condition of that? Does the accumulation of lactate after anaerobic exercise like harm our body or like cause inflammaging?
Yeah, this is a question I get asked quite a lot. So I go back to the distinction between acute and chronic response. So in exercise, there is, of course, an accumulation of lactic acid that leads to the usual response that we know of our muscles. But this is an acute accumulation of lactate, which is resolved within normally 48 to 72 hours. And so we return to an homeostatic muscle response. However, in cancer and autoimmunity and other chronic conditions, there is a chronic accumulation of lactate. So if you go and measure lactate in the tumor microenvironment, you will find a concentration of between 20 and 40 millimolar of lactate, which might correspond to a pH of about 6, between 6 and 7. In the arthritic joint, if you measure lactate, we have measured that directly, but others had done that in the 70s even. You will find a concentration of lactate between 10 and 15 millimolar, which corresponds to generally to a pH of around 6.9 to 7. So this is a low pH compared to the neutral pH that you find generally in tissues. Considering that the normal levels of lactate in the blood of a healthy person are below 2 millimolar, are between 1 and 2 millimolar, you can see that this is an accumulation of 10 to 40 times, depending whether we are in an autoimmune environment or in the cancer environment. And this is chronic. And so what we have to study is the response of immune cells to this chronic accumulation of lactate in the microenvironment. And what we've shown as a first response is that immune cells that enter the inflammatory site or the tumor microenvironment as a response to lactate remain entrapped in the inflammatory site. We have explained that by showing that exposure to high levels of lactate causes an influx of lactate in the immune cells. This causes an inhibition of glycolysis, which is required for the movement of immune cells. So this inhibition of glycolysis as a response to an exposure of high levels of lactate causes the entrapment of the immune cells in the inflammatory site. So we have proposed is that if you block the lactate signaling or the lactate sensing, you can achieve a reactivation of the migratory ability of the immune cells, which will be able now to egress from the inflamed side, which will result in a reduction of inflammatory immune cells in the microenvironment. The second, we have also shown that in response to lactate, there is an increase in certain inflammatory cytokines, such as IL-17, but also interferon gamma, we have shown, and others, which are subject of current studies in our lab. And so by blocking lactate signaling or sensing, you can reduce the production of these inflammatory cytokines and in the autoimmune microenvironment. And this is again advantageous for the resolution of the disease. We recently also shown that if you block this lactate sensing or signaling, you promote immune suppressive responses. Now, the key thing is that, and we have discussed that in a review that was published in Nature Reviews Immunology in 2020, is that this response to lactate could be tissue specific. And so we and others have shown that in inflammatory site, lactate promotes inflammation by the mechanisms I've just shown you. But a lot of literature has shown that in the tumor microenvironment, lactate promotes immune suppression, and then again becomes a mechanism that promotes tumor growth. right? So in both diseases, context, tumor or autoimmune diseases, the presence of lactate is bad for the disease, and you want to either block the production of lactate, block the signaling of lactate, or block the sensing of lactate in both cases. But the thing that we need to understand is that in both this happens in both disease contexts, but for opposite immune responses that are achieved. And how this is achieved in the different tissues that we look at is not clear at the moment. And I believe people will try to understand that in the near future.
Yeah, definitely. So in your paper, I've noticed that you have been working on the lactate transporter inhibitor. Is it a very promising way to block lactate? And is there any clinical trials that have showed trials?
I think that's again a very good question. So far, there have been only trials for MCT1, which is also known as SLC16A1. So this is a constitutive lactic acid transporter. Yeah. And this has been targeted with the use of a small molecule in melanoma. Now, the trial returned beneficial effects to the disease, but a lot of toxic effect to the patients. So the trial was not approved by FDA. And this is the common thing. We need to identify more selective molecules that act only in certain contexts. So in our case, but in the case of other investigators I know, we have focused on a lactate transporter that is only expressed by certain immune cells and in certain tissues. And we believe that would give the beneficial effects without taking away, without causing much trouble to the patients. I mean, this is a general, I think, route that the science field is taking. Like we have blocked so far systemic major signaling molecules such as TNF and interleukin-1. But the field is moving to much more specific molecules such as targeting PD-1 or targeting CTLA-4. These are checkpoints that are only expressed in certain contexts, in certain tissues at a certain time point of the disease, right? And that's, I think, where the field is moving, trying to identify more selective and specific targets that will give you a beneficial effect without having massive toxic or side effects for the patient. So that's the direction we have taken with the lactate transporter we have chosen. And I believe that we are developing that well. It will take time, but we have interest from we've had interest from a number of pharma so far, and hopefully we'll be signing a major collaboration in the near future to develop this drug.
So blocking lactate is like suppressing the bad side of inflammation, but it has come with a risk. So on the other side, how about like promoting those good ones? I've seen that in one of your papers, you mentioned interventions to slow immunosenescence, like supplementing prebiotics or like polyunsaturated fatty acids and the short-chain fatty acids. So how can these interventions be practically implemented in our daily life? Like, for example, a yogurt.
I mean, as I said, any food that contains omega-3 fatty acids, so these would potentially, together with moderate exercise. So when I say moderate, I refer to something different from strenuous exercise that elite sports people do, right? So moderate is like go for a jog, do yoga, 20, 30 minutes every other couple of days. This, together with certain healthy diet choices and integrating your diet with omega-3, which is like eating more fish or eating more walnuts and so on and so forth, could per se have a beneficial effect on the immune system, like calming down the sort of inflammaging response that we see with age, and return the immune system towards a more sort of homeostatic type of response, which is beneficial for the body. So yes, yogurt containing walnuts, for instance, could be a good sort of food, I think.
So these are all like dietary interventions. On the other side, there are like pharmacological interventions. So for these two interventions, dietary one and the pharmacological one, which one is more effective? Or put in other words, like, do they target different group of people?
Yeah, I believe you will have to target different group of people. And I believe while you are within a spectrum of health, diet and exercise are what I would advise. But when you are in a disease spectrum, then perhaps drugs become required in addition to diet and exercise, right? They become required. And as I said, in terms of immunosenescence, drugs that perhaps could help, I think there is enough evidence out there that metformin has an effect on immunosenescence. But I believe other new drugs like GLP-1 inhibitors and SGLT2 inhibitors could be new drugs that will be shown to have a beneficial effect on immunosenescence and inflammaging.
So you have also mentioned metformin several times in this speech. Like in terms of the future research on inflammation and aging, which target do you think holds the most promise? Like those like drug targets or any kind of other interventions?
Yeah, I will not go into very specific response here. I mean, I have in mind a couple of targets, but hopefully we will publish those soon. But I believe that what promises to deliver new important drugs are key metabolic enzymes, as well as transporters of metabolites, such as amino acid transporters, glucose transporters, lactate transporters, and so on and so forth. I think these are two key classes of targets that will deliver important drugs in the not so distant future, and are already delivering, such as, as I said, the GLP-1 inhibitor and the SGLT2 inhibitor, that are two very novel drugs.
So after listening to your sharing, our audiences have lots of questions. Can I hand these questions to you, Professor?
Yes, I can try to answer.
So the first one is about arthritis. So arthritis is also caused by chronic inflammation. Effective ways to treat rheumatoid arthritis.
I mean, I think the drugs that are available at the moment are good drugs, of course, blocking TNF, targeting B cells, methotrexate, and so on and so forth. Unfortunately, not all patients respond to those. They perhaps treat more the cause of the disease, but not the worst symptoms of the disease, which is chronic pain. So I am aware that in UK, for instance, there is a lot of investment into paying research in autoimmune disease, because, of course, an anti-TNF like infliximab will not cure all types of pain that are caused by rheumatoid arthritis. So yeah, there is a lot more to be done there. Besides these drugs that are available, such as anti-TNF or rituximab, that targets B cells, they target very broad mechanisms, as I said, and they come at a cost with partial immune suppression, increase infections in patients that take them. So yeah, the field has to move on in terms of rheumatoid arthritis and arthritis in general, I think.
There has been a drug called Senolytics that has been widely discussed because it can clear out senescent cells. Can these kind of senolytic drugs be implemented into treating inflammation and aging?
Yeah, definitely this is a field that I haven't directly worked on, but of course is a big field out there. These drugs clear senescent cells, so they should provide a benefit to inflammaging and to the diseases of aging. But I believe trials will need to be conducted to get to the bottom of it. There have been promising publications out there in the preclinical space, early clinical trials, but we will have to see what they deliver in the near future.
So the next question is about daily examinations. If we want to detect our inflammation level in our daily life, which indicators we need to pay attention to? What are those indicators?
Well, yeah, then again, there are, of course, broad markers such as C-reactive protein, interleukin-6, TNF, and so on and so forth. But the problem is these are systemic indicators of inflammation, but they don't really tell you whether specific mechanisms are altered. For instance, in obese people, when they lose weight, I believe these systemic inflammatory markers go back to a sort of normal level for a period of time. And yet, there are publications out there showing that the innate immune response is still programmed towards inflammation. And our own data show that adaptive immune response does so as well. So even though your systemic inflammation indicators of inflammation have kind of returned to a roughly normal level, that doesn't mean that you have corrected the damages caused to the immune system by the obesity.
So one of our audience also asked a question about the innate immune disorders. He's wondering whether the innate immune disorder is related to chronic inflammation. What is the relationship between these two concepts?
Well, innate immunity is a major driver of inflammation, right? Macrophages are a major, and neutrophils are a major driver of inflammation and are the first cells that enter the inflammatory microenvironment, so that they play a major role there. Then later on during the response, you get the activation of the adaptive immunity, and that cooperates with the innate immune cells to propagate the inflammation, right? Which, unless it resolves, then it becomes chronic. So yeah, they play a major role.
Curious about the relationship between the chronic inflammation and the endoplasmic reticulum stress. So I've heard that their researchers suggest that NDA deficiency can lead to the glucose metabolism to release ROS, thereby downregulating the GPB. And they can result in this kind of issues. So what is the relationship between these two concepts, the chronic inflammation and the endoplasmic reticulum stress?
I mean, funny enough, the first paper I published in my career in 2002 was on the on NFκB signaling via the endoplasmic reticulum. So there is a strong link between the endoplasmic reticulum and the inflammatory response, both as an input. So the inflammatory signals require the endoplasmic reticulum, and then the downstream responses of an inflammatory signal also require an endoplasmic reticulum response. The endoplasmic reticulum is a key organelle in the cell that leads to the correct folding of proteins, right? And that is key to produce a correct protein that then it's like for inflammatory factor is released in the system. So there is a huge literature about how inflammatory cytokines or inflammatory proteins of the cell are linked to the endoplasmic reticulum via, for instance, the unfolded protein response. The acronym of that is UPS, and then that leads to the output of the inflammatory response, which is cytokine produced. So yeah, that's a key aspect. And there is a lot of literature nowadays of endoplasmic reticulum unfolded protein response in the immune cells.
And the next question is about body temperature. Someone is curious that is body temperature related to inflammation? Does a high body temperature means that we have a higher risk of inflammation?
Okay, well, I mean, of course, interleukin-6 is a major controller of body temperature. And so, for instance, in fever, you have an increase in interleukin-6 that leads to increased body temperature. And that's a signal, a danger signal of an inflammatory response happening because a pathogen has infected us. Whether increased body, like chronic increased body temperature is linked to inflammation, it's not something I have dwelled about, so I wouldn't go into that, sorry.
That's fine, that's fine. It's just some like so well, when we come back to food, there are in our daily life there are food labeled as like pro-inflammatory or like anti-inflammatory food. Like milk has been labeled as a pro-inflammatory food. And then how about carbohydrates? Are they like pro-inflammatory food?
So I mean, I will let's say that in very broad terms, whatever promotes anabolism will promote an inflammatory response, and whatever promotes catabolism will favor an immune suppressive response. So yes, carbohydrates will, in theory, I mean, in very broad terms, right, will are at risk of promoting inflammatory response. Generally, we eat all sorts of things, so it's a balance. But yeah, I wouldn't enrich our diet with omega-3, olive oil, and all sorts of polyunsaturated fatty acids.
You have mentioned that the fish oil is also one of the thing that can fight an inflammation. And one of our audience is taking high dose of fish oil capsules containing EPA. He's wondering whether it is a good way to kill inflammation.
There are definitely reasons to do that within the scientific literature. We have published papers where we have given to mice a diet enriched with fish oils. We have treated immune cells with EPA or DHA, and we've shown that they have an effect on the immune response. So there are reasons to do that. The problem is that our body, it's a complex machinery, right? So perhaps in certain people, taking EPA will not be sufficient. I mean, if you are an overweight or obese person with type 2 diabetes, probably taking EPA will not be sufficient to cure you.
It seems that the time is running out. We're not willing to, but our interview is coming to an end. Thank you again for sharing so many important informations to us and sharing all your insights on this topic. And I hope that, Professor, that we can get in close contact in the future and wish everything goes well with you.
My pleasure. Thanks for inviting me. It was lovely to, address your questions, and hopefully the audience have found them, interesting. And thank you for your time.
Thank you so much, and looking forward for future interactions. And I may end the call today. Thank you so much, Professor, and
Bye-bye now.
wishing you a good day. Bye.
Bye.
Bye.